Welcome, ladies and gentlemen, to Berlin Brains, the joint event series of the Urania Berlin and the Charité, in which neuroscientists present themselves and their work. My name is Jochen Müller. I have the pleasure to be your host this evening, and I welcome you to Modern Neuroscience in the oldest science center of the world. Berlin Brains is in the 8th season this year, and this year presented from the Einstein Center for Neurosciences, the Excellence Cluster NeuroCure, the MDC for molecular sorry, messed it up right in the first minute. Max-Delbrück-Centrum for Molecular Medicine in the Helmholtz Association, the Bernstein Center for Computational Neurosciences Berlin, and today the SFB 1315. This is a collaborative research center focusing on mechanisms and disturbances in memory consolidation from synapses to systems. And it will become a bit clearer today what this means, because in Berlin Brains, we present science of the brain, the nervous system, its cells and functions in a way that is understandable to everyone. Well, when you look at the cells of the brain and what they do and how they communicate with each other, often one question remains. How does their activity lead to us perceiving the world outside? How the hell does this work? And during the next hour, Clarissa Whitmire and Jean-Sébastien Jouhanneau from the Neural Circuits and Behavior Group of Professor James Boulay at the MDC will talk exactly about this question. They will talk and explain the most, I would say, underrated of our senses, somatosensation, the sense of temperature and touch, among others. About how the brain transforms my touching this into me perceiving the cotton of my t-shirt. But they will not give a boring scientific or philosophical presentation with a lot of tech terms and graphs and tables, but they will do this in an understandable way, and we hope so in an enjoyable way, and I'm quite sure you will do that very well. Because this is what we do in the Urania. We hope you enjoy it, we hope you learn something from it, and we hope that you want more from it. Come here to Schöneberg and visit us, or if you watch from at home, go to urania.de, which you may have already done, otherwise you couldn't watch us, and where you can also watch videos from former Berlin Brains events, because since 2020, we stream the events live, we record them, and we put them on the Mediathek of the urania.de. We also show them on YouTube and www.dasgehirn.info. For example, in March, we talked here about protein origami and how misfolded proteins damage our brain. Today, we take a cold, hard look on how the brain represents temperature. You can also get in contact with the speakers, ask them questions. I will tell you in a bit how this works. But if you really want to get in contact with them, really discuss with them, have a drink with them at the bar, you need to come to the Urania here in Schöneberg. And maybe you can do this in June when we talk about how a two-phased protein called tau does something very important for us in health as well as in disease. So come here and enjoy science communication at its best, I would say. I gave you these two examples of March and June because the topics are quite straightforward. Of course, talking about clinically relevant topics is always interesting, right? How to understand a disease, how to cure it, quite straightforward. But we all too often forget that clinical or applied research bases on basic research. And this is what we will focus on today. We found two persons working on one of the questions that is among the oldest of mankind. And only now methods become available for them to maybe get an answer for this question. And since they both work together in the same group using similar techniques, we changed our conceptual motto a bit. Normally, we have the conceptual motto, junior meets senior, means there's the group leader or PI, first given an introduction to the field, and then mostly a PhD student gives you a bit more detail about one project. But today we have two postdocs, two people who have their PhD already, but are not group leaders, but they work on the same project or a similar project, so why wouldn't they present together? So today, they both share the stage and they will take turns in speaking. We will do this the first time, so please give us feedback afterwards. It's also only the second time we do this show in English. Please also give us feedback on how you like it. I said that you can also get in contact with us. Afterwards, we have a Q&A session. So the talks will roughly be 45 minutes. Then I come on stage. You can ask questions. For you here in the hall, when you want to ask a question, please go to the microphone and talk into the microphone so that the people in the hall can hear you and also the people watching from home. You guys watching from home, you can also ask us questions. Just open your mobile device, open the browser, go to slido.com, SLIDO.com, and enter the hashtag of today, which is BB0522 for Berlin Brains, May 22. Slido.com, BB0522. Type in the questions and I will read them on my mobile device and ask them. And then you will hear their answer. So now let's see who we will be hearing today. Normally I ask the people, give me a short CV and tell me why you are here, why the work fascinates you, and maybe tell me one or two hobbies so that I can present not only the researcher but also the person. These two guys, they wrote me a letter that was so touching that I thought I can't just sum it up. I have to read it to you. It was too long to read all of it, but I will read parts of it because I want you to be as touched as I was. So first, Clarissa Whitmire. She grew, and I will read if I was her. I'm Clarissa Whitmire. I grew up in Apex, North Carolina, with 3 brothers. I spent my summers lifeguarding and enjoying the sun. In school, I was particularly interested in mathematics, drawn to equations that could explain complex systems. It wasn't until I went to college that I found my passion for neuroscience. While studying engineering, I took a course on bioelectricity and created models of the heartbeat and how the electrical activity could synchronize the heart to pump. When we moved on to the electrical activity of the brain, I was fascinated by the way our neurons use electricity to communicate. At this point in my career, I knew absolutely nothing about graduate school or pursuing a career in science. I had worked in a number of different jobs and planned to continue on the industry path. However, my bioelectricity professor introduced me to the academic world and to basic research. And since that moment, I've been consumed with a third search for basic knowledge. I'm really interested in some very basic questions. For example, how can we actually see the world around us or touch it? It is important to understand that perception is a construct. As Paul Bach-Irita put it, you don't see with the eyes, you see with your brain. This is my driving principle. I want to understand how that percept is constructed. Why I participate in Berlin Brains? Because I think it's important to inspire people who may not otherwise have access to regular scientific discourse, like my younger self. I'm the living proof that everyone can become a scientist. All you need is to be curious and maybe a bit passionate. And if I fall flat, at least I hope I've entertained you with some neuroscience. What free time? I like that question. And I love the answer. I'm in the Berlin Spielplatz scene with my 2 kiddos. If it counts, my favorite hobby is traveling. I love to experience novelty, learning something new, whether it's a new food, a new phrase, a new perspective. I really take learning in every form. I crave novelty. Other hobbies, I love reading quickly, I love cooking slowly, and when relaxing, I love telenovela-style drama shows. This is Clarissa Whitmire. Who is Jean-Sébastien Jouhanneau? He grew up in Tours, in France, and I will also read as if I was him. I was interested in how things work very early on. I would unbuild things and mostly not being able to put them back together until one day I managed to fix an electronic toy that was very important to me, and that was when I discovered the power of knowing. When you know how things work, you can fix them. During my high school years, I had a wonderful teacher, Madame Magin. She was 65 years old, but she had still that passion to teach. I really discovered biology in her class. I was in Wonderland. I decided to become a scientist. I wanted to learn more about the brain. So I visited the University of Tours in the center of France for my master's degree. Under the guidance of Professor Le Genec, a well-known electrophysiologist specialized in heart cells, I got to know electrophysiology, and this blew my mind. After so many years looking and learning from cells, I was able to talk to them. To understand the brain, I went to Bordeaux to join a lab working on Parkinson's disease. There I learned about how brain regions interact to generate a controlled movement. I used electrophysiology to assess the properties of neurons from these different brain regions. Afterwards, I was convinced to better understand the brain, one should focus on how it's built. So for my PhD, I joined the lab of Professor John Isaac in Bristol, UK, and then Bethesda, Maryland, United States, specialized in brain development with a focus on the somatosensory system. My PhD helped me to understand how communication between neurons during development was modulated by incoming sensory inputs. This is important during maturation, but also in adulthood, generating brain states of activity. To better understand the role of these different states of activity on neuronal communication, I then joined the lab of Professor James Pullet here in Berlin. He was famous for his work on neuronal mechanisms of switching brain states like wakefulness and quietness. Here I'm combining to photon microscopy and electrophysiology. We are investigating neuronal communication in the living brain. When I'm not in the lab, I do martial arts since the age 12. I started for karate and now do kendo. And I love everything about it, the Japanese culture, the etiquette, the simplicity, the respect, and the attention to detail. But my main driving force is my family. I have a wonderful and supporting wife and 4 amazing kids who I'm very happy to welcome here in the audience tonight. They are the light keeping me focused and driven. Their creativity and energy is inspiring. My daughters and my son are 13, 15, 16, and 17 years old. They are growing so quickly that when I come back from conferences, I can notice it. When I see them, I know I'm fortunate. I would say there's little more to say. Let the rest have themselves. Please welcome with a warm, very warm applause, Clarissa Whitmire and Jean-Sébastien Jouhanneau.
Thanks, Jan. Thank you. All right. Okay, well, thank you very much, Joran, for the introduction. Thank you for the invitation. It's a real pleasure to be here and an honor to be able to talk to you about our science. As Joran put it, I really like from the beginning to actually build stuff and rebuild stuff and mainly failing. And actually, that's what I'm still doing, right? To understand the brain, what we do is trying to remove a part of it, a part of the network, and understand the function of it. And doing this, we actually increase our knowledge of how the brain is working. So today, we're going to talk about how the brain represents temperature, right? And when we say represent temperature, the word represent means where in the brain this sensation, this touch or temperature sensation is actually activating cells in our brain. That's what we call representation. So where in the brain cells respond to this particular stimulus. Okay, the hypothesis is that this is in the somatosensory cortex, right? As Joran put it, somatosensory is all that refers to the skin, which receive this type of stimulation, the touch, the temperature, the cold, the warm. Right, that could mean, I mean, it might be trivial for you as a question, but actually we're going to talk about 2,500 years of research, and showing you that it's not actually that simple. And actually, as today, we don't really know everything about it, and the principle of how these neurons actually help our brain to represent the surrounding world. So, yes, it's not easy, and the problem is that it's very complex. The brain itself is very complex. Take the heart, for example, right? We talked about the heart before. Well, you just open it, you look at it, and you realize it's a pump, okay? It's a big muscle. It's got 4 chambers, and you know that its role is to actually pump the blood to give nutrients and oxygens to the organs, right? If you look at the lungs, for example, and you open them, there are bags, 2 big bags, which will allow the oxygen from the air to be transmit it to the blood and oxygenate the blood for the body to be able to live. But if you look at the brain, right, it's like 1.5 kilograms of fat. Right? You open the brain, well, you can't really see anything. It's just a big ball of jelly. It's really complicated. The brain's got 86 billion neurons. It's 86 with 9 zeros. It's quite a lot. Right? And if of this neuron, then the brain cells, they receive thousands of inputs from the other brain cells. Right? So it's highly complex to understand how the brain works. And that's why today we don't really know or understand everything about the brain. So you have to see it as a big puzzle. Right? The brain is a big puzzle, and to understand the brain, we have to put every piece of the puzzles together. But actually, there is pieces that we actually don't know about yet. Right? It's so complex that there might be pieces that we'll discover in 10, 20 years. In our case, we choose one piece of the puzzle, which is how the brain represents temperature. Okay? And actually, I would like to start by experiment. Okay? So, it's a little experiment. If the answer is yes, you will raise your right arm. Okay? If your answer is no, you will raise your left arm. And we'll do that all closed eyes, okay? So you ready? You can close your eyes. All right, everyone close the eyes, okay? You remember, right answer is the right arm, and the no answer is the left arm. Okay. Can you hear me? Now you have to raise your, yeah, exactly. Right, that's the game, okay, you can hear me, very good. Now, can you touch your nose, hold on a second, can you touch your nose with your left index finger? If you can't, left, okay? Okay, you can, okay. Right, very good. Now, last question. Can you see me? Well, of course you can't, right? And that's the end of the experiment. You can open your eyes, right? No, but that's true. I mean, you can't see me because you need your eyes to actually see me, right? And that's the basis of neuroscience, right? So we've got a brain, it's 1.5 kilogram of jelly, and it receives sensory information. And these sensory information are transduced from the periphery by electrical impulse to the brain. And as we just saw, we need your eyes. The eyes will actually, the light will activate the retina, and the retina will send information to your brain. Right? Your nose will be able to sense odors, activating the olfactory bulb, and then sending this information to your brain. The same for the mouth. The tongue and the taste bud will actually receive the sensory information and will transmit it to the brain. The hearing, right, the inner ear will receive hearing information and send it to the brain. And the same for somatosensation, right, which will be reaching the brain and actually reach this area, the pink area upstairs there, which is the somatosensory, the primary somatosensory cortex. Okay, so as I told you, we need our eyes to see. And I want to make a point there, right? This is a picture of my last vacation, Hawaii, right? Actually, that's not, I mean, that's Hawaii, but that's not my last vacation. It was maybe France, but that's, let's say it's a really beautiful scenery. What you can see there is something that is beautiful, right? You can almost start to feel the breeze, right? You might smell the seawater, okay? You might actually see the color of this sunset, okay? But actually what your eyes are seeing is just a mix of color, which are only 3 colors, right? We actually perceive only blue, red, and green. So that picture I showed you, you see a bit of blue, a bit of green, a bit of red, and then this goes into your eyes, to your brain, and that's how you reconstruct that image to actually make this percept of that beautiful beach in Hawaii. Okay, so we need our eyes to actually see what's going on, but it's our brain that makes us see our surrounding. So now, let's go back to our question, which is how the brains represent temperature. First of all, I wanted to say that I can prove it to you that the brain reconstructs images, right? And I will show you this image now, and you will have to tell me what you see, okay? So I show you this, and you tell me what you see, or maybe you tell me what you saw first, because indeed it's quite known that there is the wife in this image, but there is also the mother-in-law. All right? And so our brain actually see what it wants you to see. It's reconstructing the image based on your experience. Interestingly, there is a study saying that younger people will tend to see the wife first, and older people will actually see the mother-in-law first, right? So our experience, which is very important in how we perceive the world, can actually affect the way our brain reconstructs our surrounding world. Okay. Now, go back to the question of how the brains represent temperature. It's a key question in neuroscience. Here you might see a beer bottle, right, or a mug of coffee. But actually what's important to you is to know if the beer bottle is cold enough or the coffee mug is still warm enough to be drunk, right? So that's the very important question, and that's what we're going to talk to you about today. But first of all, let's dive into this history of science to see how we evolve on that role of the brain in perceiving temperature. Right.
Okay, so let's go back about 2,000 years and see what we think about this. So these questions, like Jean-Seb brought up, of we see with our eyes, it seems very basic, right? We're like, of course we see with our eyes. But what our skin actually senses has been a subject of debate for thousands of years. So Aristotle is actually sort of credited with this idea of having 5 senses, although it was probably very much common sense back then now as it is now. But he specifically in somatosensation postulated that somatosensation, or the skin senses, is a plurality of senses. It's not just one thing, it's multiple things. And this idea was expanded upon up until about the 1st century. And we don't really have a recording of what they thought the senses actually were, but they did believe that there was more than one. But then, for about 1,000 years, 700 years, 500, 700 years, this changed. People started to believe that actually the skin was sensing only a single thing. There was only one sense for the skin instead of this plurality. So actually for hundreds and hundreds of years, we thought the skin was just doing one thing. And it actually wasn't until a really prolific anatomist or physician or scientist, however you want to call him, Avicenna, came about in about the 11th century, that he renewed some of these ideas that touch or somatosensation was a plurality of senses. And similar to his predecessors millennia earlier, he didn't exactly dictate what all of them were, but basically the idea was that the skin could sense as many things as there are to sense about an object. So 2 that were recorded at the time actually was pain, the idea that pain or pleasure could be encoded by the skin, and another was actually hunger and thirst, that your skin was encoding this information. And these ideas about what was actually being encoded by the skin continued to expand. So this is just a subset, but it was believed that skin would sense humidity, so moisture versus dry, pain versus pleasure, temperature, hot versus cold, hunger versus thirst, or light versus heavy. But again, this is just a subset. So there was tickle and titillation and all sorts of things that were believed to all be encoded by the skin. And up until this point, essentially a lot of what was being done by philosophers or anatomists. It was all observational or theoretical because we really didn't understand how information was being encoded by the skin. And in the 18th century, there was a really interesting observation by Erasmus Darwin, who was the grandfather of Charles Darwin. So some say that he was overshadowed by his grandson and then wiped out of history. Otherwise, he was actually quite prolific in his own right. But just so you know, I'm going to rearrange the timescale here. We summed up the first 2,000 years quite quickly, and now we're going to focus on the last 250. But what Erasmus Darwin did is he made an observation. One of his cousins was a physician, and he had a patient. And this patient couldn't feel a sharp poke to the foot. There was some issue. So when he poked the foot sharply, poke, 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 no feeling, many spots. However, he took a hot poker out of the fire that was burning hot, and he brought it really close to the skin, and the patient could still sense that. The patient could actually feel the heat, but couldn't feel the what I would call pricking pain, which is like a painful, sharp poking. And this provided sort of a really nice piece of evidence that temperature was a sense in its own right. That temperature was something the skin was encoding that could be independent of other things, like a painful prick. And so this, I would say, is sort of the start of when people started to do more careful experiments, let's say, on somatosensation.
Yeah, and that particular experiment there, where Lucia and Luigi Galvani actually were working on a topic at that time called medical electricity, right? And the idea was that there was electricity in the body. They thought that the body can generate electricity, all right? And they did this kind of experiment using electrostatic electricity to actually probe for where the electricity comes from. So the story is that as they were preparing the frog, the dead frog, and they were actually dissecting the legs of the frog to reveal the muscles and the nerves, Lucia was actually rubbing the skin, trying to see if that was going to give some static electricity. And one of the assistants there, which was still prepping the mouse, the frog, sorry, used a scalpel blade with its metal scalpel blade, and then which probably get the charge of that the static electricity, and while touching the sciatic nerve of that frog, the leg started to move, right? So as the leg was alive again, and that was stunning. That is actually a turning point in sensory neuroscience, because this experiment, right, showed us that electricity can be actually conveyed by this nerve. Of course, they were wrong in the sense that they thought the electricity was within the nerve, and Volta, Alessandro Volta, at the same time actually proved that it wasn't the case. Nevertheless, these nerves can actually propagate electricity, electrical impulses, which is, again, a key point in this. And that's what they call bioelectricity. Right.
Yeah, and so like Jean-Seb said, this was pivotal. Before this point, it was thought that essentially everything you sensed, some copy of that was actually in your brain. So if you saw something red, there was somehow some copy of red light in your brain. This was saying something fundamentally different. And that was really formulated by Johannes Müller, who here in Berlin formulated what was known as the law of specific nerve energies. And so following up on the Galvani's work, he found this. This is a translation from his original work in German. I apologize for my lack of German. But he said that electricity can simultaneously affect all sensory organs, and yet every sensory nerve reacts to it differently. So one nerve perceives it as light. So what does that mean? It means that if you stimulate the optic nerve, you'll always get a visual percept. You'll never stimulate the nerve connected to the eye and hear something. That's not what happens. There's a specificity. But he goes on to say another hears it as sound, another one smells it. So he developed this idea of specificity within the sensory pathways, that while they all are sensitive to the same thing, in this case electricity, the percept that they develop is dependent on which sense organ they're connected to. And so if we return to sort of our neon man, what we can say is that now these nerve fibers can be tracked, right? And, there was a lot of ideas of people expanding on this hypothesis. And so Helmholtz is really famous for his work in the visual pathway, diving down into the color specificity that we talked about already, that he started to separate vision into multiple independent pathways. But in somatosensation, there was some really seminal work that came about 50 years later. So there were actually 3 scientists, Magnus Blix, who was in Sweden, Alfred Goldscheider, who was in Germany, and Henry Donaldson, who was in the United States. And what's particularly interesting is that all 3 of them discovered essentially the same thing in the same decade, actually. So following up on Johannes Müller's work, they actually experiment on themselves. So a lot of the work in this time, they will just take their hand and try different things. So they started actually by taking electricity and stimulating their hand and saying, what does it feel like? So under the knowledge that all of these nerves are electrically active, if you stimulate them, what do you perceive? And the idea that they were going after is what are the different levels of touch? What are the different levels of skin senses? Just like we talked about before that we've been asking for over 2,000 years. if I stimulate one space, what does it feel like? And what Magnus Blix found is that when he stimulated different spots, he could get fundamentally different percepts just depending on where he was in the skin. So stimulate one spot, it'll feel like pain, another spot touch, another spot cold, another spot warm. And so he decided to explore this a lot further in the context of temperature. So he developed this device that could be either warm or cold. So essentially in this device, what you see is there's sort of two pots and containers, yeah? One has hot water, and one has hot water, and one has cold water. And depending on how you level those, changes the temperature of the thermode. So this right here is the thermode. And what he would do then is he took his own hand. If you look at all of his original drawings, it's like back of left hand, inside of left wrist, right cheek, because people just experimented on themselves. And he would take this thermode and touch all these different spots on his skin, just touch, 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 and he would record what does it feel like. And what he found was really fundamental for the thermal pathway, actually, and I would argue still something we test to this day. In fact, we tested it just last week at Lab Retreat. That specific spots in the skin were sensitive to either warm or cold, and that these spots were discrete. So what he found was that within the skin, you could find a single spot on your tissue that would respond to a warm stimulus but not a cold, or a cold stimulus and not a warm, or nothing. Also, it might not respond to anything. So it really discretized our view of somatosensation. And so what I've shown here is all the work from Magnus Blix, but Alfred Goldscheider and Henry Donaldson really expanded on this work. They tried a bunch of different ways, and everyone found the same thing, that there's these warm and these cold spots in the skin, and that's likely how we're sensing temperature. So now if we have some understanding of how the thermal information is encoded, let's say, by the skin, how it's represented by the skin, we start to ask, what does this mean for our percept?
Well, actually, I've got a question there. Do you think that you can trick the brain with different type of temperature, like we can trick the brain with different illusions, visual illusions?
Yeah, so actually, thank you, Jean-Seb. That is exactly what people wanted to do. So there's a really cool illusion in the thermal pathway that was developed in 1896, so not long after these hot and cold spots were developed, that they said, can we stimulate them at the same time? And what will happen? So in this case, they took copper coils and ran hot water and cold water through them. The hot water alone is just warm, and the cold water alone is just cool. But when you spatially interleave them, what you actually get is a percept of pain. It can be incredibly painful. And this is now called paradoxical heat, or the thermal grill illusion. So even though what's encoded at the skin is not painful, the reconstruction within your brain is that of pain. And so also for 2,000 years, for over 2,000 years, we've known there's a tight link between temperature and pain. We all use a cold pack when we get an injury, right? It's an analgesia. We also know about ice cream headache when you drink your milkshake too fast. There is some link between temperature and pain. But there's other illusions beyond the pain. Sorry. Another one is where you can actually check the spatial summation of the thermal pathway. Thank you. And if you warm just your two outer fingers like this, what your brain actually perceives is that the middle finger is also warm. This is called thermal referral. Thank you. Sorry, I'm choking up here. Thank you. And in thermal referral, what you can get is an illusory percept of warm without any warm stimulus actually being applied. And while this example was demonstrated first in the fingertip, there was a really interesting study where they tried to separate warm from touch. And when they did this, they used IR light to warm the skin. And so basically, they sat people in a chair, they shined an IR light at one side and an IR light at the back. And they just asked the subjects, did I warm your chest or your back? Seems like a straightforward question. If someone touched your chest or touched your back, you'd never get that question wrong. But in the case of temperature, people got it wrong 15% of the time. So it's pretty astounding, actually, that the thermal pathway is so different, even though it's also encoded in the skin, just like touch. Sorry. And you know, these are kind of hard for you to test out here because we don't have a lot of thermal stimulators, but if you'd like to try another thermal illusion, or another tactile illusion, somatosensory illusion, there's one called the dead man's finger that I learned about. So if you want, you can try this with the person next to you. You put your hand up like this, okay? And then, can you hold this? Oh, yeah, okay.
No.
And you take your other hand and just feel up and down the finger. And it feels like you're touching a dead finger. Do you feel that? Okay, if you're not around someone, then you can't feel it. But so basically the idea of that is that it's a proprioceptive illusion, where you're expecting the feedback from your finger as you touch it, and when you don't feel it, it feels like you're touching something that's completely abnormal. Yeah. So anyways, we can also do this at the bar later. I'm really into illusions, you'll see from this, so I'm happy to chat more about it in detail. Right, so all of this is just to say that the percept that we have is not always representative of the actual world, and that our brain is creating the construct that we experience. And in the thermal pathway, this has been tested in quite a few ways, I would say. Okay.
And it's actually only in the 90s that we really figured out, I mean, I would say we, but actually it's David Julius and Artem Pataputian, which received actually the Nobel Prize for that in 2021, last year. So it's only in the 90s that we actually found which proteins sense heat or cold. So the way they did that is that using capsaicin, we know from, you all know from your experience, that when you eat spicy food, you have this burning sensation. And we actually know that these burning sensations come from capsaicin. But what we didn't really know at the time is how this capsaicin could transduce these informations to the brain, right? And at that time, so David Julius found out that it's actually this receptor, the red one, the TRPV1, that was responsible for transducing this electrical impulse when he was in contact with capsaicin. And then they also found that TRPM8, the blue receptor there, was responsible for transducing cold sensation, right? For this one, they use menthol, and menthol is actually activating TRPM8 receptor to transduce cold sensations. This receptor, they actually express into these fibers that you can see in this schematic, where you see the blue fibers going to the blue spots, referring to what Clarissa said just a minute ago about this warm spot and cold spot. Right? And also there is a controversy about maybe some fibers expressing both of these receptors, which would transduce some kind of mixed informations to the brain. Okay, so this was a very critical point as well in neuroscience, because at this point we could go further in understanding how the brain represents temperature. So we have these two receptors, right? Now the questions remain, where in the brain neurons can be activated to actually represent temperature, right? Our hypothesis, I told you, is this pink area there, which is called the primary somatosensory cortex, and receive sensations, information from the skin, right? That we know of. So that's our hypothesis. And to actually probe for this, we use new technologies, and that's how it works to push the boundaries, the frontier of our knowledge. We actually use new technologies to reveal new properties or knowledge that will help us understand the brain. Okay. But to do this, we actually need an animal model, because we want to know at the cellular level. So while we can talk to humans and ask them what they feel, right, we can't really modulate or manipulate their brains. So we use, in this case, the mouse model, right? The mouse model is very important to us because it's a mammal, for example, for starter. So it's we're both mammals, humans and mice, and they have the gross same organizations, meaning that they do have nerves. That's the blue fibers you see there. They're actually peripheral nerves. That's these nerves that will convey sensory information from the periphery to the spinal cord, which is the white line you see in the middle, and these will go to the brain. Okay, this is exactly the same idea, and you can see this in these animations. If you stimulate the paw of the mice, it will go to the spinal cord and then reach the brain. Interestingly, mice got a forepaw with 4 digits. right? Like us, I mean, we've got 5, but same type of organization. So to us, translationally speaking, the model mice is very important. Another fact is that I can actually modulate or modify the genome of these mice, which is very important to us because, as I told you at the beginning, we want to unbuild, right, to understand what's actually how the brain is working. So we will use these tools to actually modify cells in order to maybe see them in action in the brain, or maybe block them so we can actually see what they're actually doing in the brain, and so on. Also, the next important things, obviously, is that mice and humans have a brain, and this brain is roughly, I would say, very similar to one another. So you can see on the left, that's the schematic of a human brain, and on the right is a schematic of a mouse brain. And what we are interested in is the primary somatosensory cortex. I told you already, it's here on the human brain, and in the mouse, it's there. Okay? So same kind of gross organizations. We can modify cells so we can actually interact with their function, and so we can start actually trying to understand how it works. I told you about the technology. It's very important to be able to push the limits and understand how these cells works. I told you about genomes and how we can modify cells. And in this case, what we've done is adding to the genome a protein that will react when the cells are activated. Meaning that each time a cell is activated, it will emit light. And for us, it's very important because that's how we will understand how the brain represents temperature. We will be able to see when the cell is activated. We will be able to see how the cells, their regional organization, right? And understand how the cortex can actually integrate these informations. Right. So, and Clarissa will tell you about what we do.
Yes, so what do we do? So like Jean-Seb said, the amazing thing about using a mouse model is the transgenics. And like he mentioned, we can make these cells fluoresce. So we can visualize what they're, we can actually see what they're doing. And because we're working in basic research, we're right on the edge of things that are being done. We can't go to Amazon and buy the types of tools that we need. Instead, we have to make them ourselves. So I spend a lot of time building microscopes and calibrating microscopes so that we can measure the activity of the neurons in the living brain. So this is a video that I made of building what we're going to talk about first, which is a wide-field microscope. So a wide-field microscope is much more similar to a camera system. It's using visible light and just recording the activity on a camera. It's quite literally taking a broad view of the activity. So when we think back to the look of the mouse brain, here we have somatosensory cortex. What we can do is actually get a window into the activity in the brain. And what we're looking at here is not at the level of single neurons. What we're looking at is integrated activity over the cortical surface. So it's a little different than saying which cells are activated. It's more saying which regions of the brain are sensitive to temperature. That's the first question we wanted to ask. So to ask that question, we used this microscope to start. So what I'm going to show you here first is a video. And this video is showing you the change in fluorescence, the change in light that we see in response to a thermal stimulus. So just to orient you, this is the same image that you're seeing here, the same orientation. And on the bottom, we'll show the temperature. And you'll see when the temperature drops, you'll see the activation in the cortex. Okay, you believe me. So here you can see that the temperature comes on and the whole brain lights up. Not the whole brain, this is the region that's centered on somatosensory cortex, but it's very clear. We can find which region of the brain is responding to the cold stimulus.
But let's go back to the single cell, right? Because eventually that's what interests us. Our understanding how the cells activate and how they organize in the cerebral tissue, and in this case, somatosensory cortex. And I want to show you this, right? Because to us, it's inspiring, right? To me, it's the symbol of pushing the limits of our knowledge. This is the Hubble telescope. I mean, I could have shown you the James Webb, but it's not yet working, so still being tested. But this telescope is actually giving us an amazing view of the universe and pushing the limits of this universe. So now this is one of the pictures that the Hubble telescope has taken, where you can see stars and galaxies, right, that we couldn't see before. And the idea there, what I want to point forward, is that we're using similar technologies, not to see stars, I mean, that's our stars, but to see cells in the brain, in the living brain, right? And what we do for that is get in touch with other labs, so we can also build ourselves, but in this case, we also went to a photonic lab in Charité, led by Benjamin Yudkiewicz, to build this microscope. So here, it's one of the microscopes we built into the lab, into James Poulet lab, where you can see that, you know, there is lots of things going on, and it's not that easy actually to build. It looks easy, but you have to assemble things together, so that's the easy part, right? But then you have to tune your microscope so that the emission path is actually going into the specimen the way you want it to go. This is the microscope itself. We call it a two-photon microscope. I won't get into the details, but if you want to know more, we can talk about it later after the talk. But this microscope, again, can enable us to see with a laser. So the laser goes into this laser path there and to the objective. And the mice, you have to envision the mice here, right? Okay. And what we're going to see there, it's a bit fast, I know, but it is cutting edge science, right? So what we did there is actually recording from multiple neurons at the same time using a technique called electrophysiology. So we can record spikes from single cell. I can give you the detail of that, but the main important thing is that we can probe connectivity between these cells, which is really important because that's how the brain works, right? Connecting between cells to actually be activated at the same time and represent the sensory stimulus. So these cells have been filled with a fluorophore, and you can see the larger bits of it in the middle. It's what we call the cell soma, and these branches there, to me, they are really beautiful. They're what we call dendrites, and that's this part of the cells that receive sensory information. Okay, and actually I'm going to show you this. That's the cell we record from. I want to make a pause there again, right? We are the first lab in the world to have been doing this type of recordings, right? And again, this is really cutting edge. That's how we can understand the brain. That's how we can push this limit, these frontiers of our knowledge, right? So again, just to remind you, this is the cell soma. That's the ball you see there, where the nucleus of the cell is, all the information the cell needs, right? And you've got all these branches, which are dendrites, which receive information. And this line there, going off the cell, the neuron, is the axons. That's how cells can communicate with each other. Okay. So now I will pass on again, and you will see single-cell resolution activity looking at these somas in the mouse brain, living mouse brain.
Right. So with the two-photon microscope that Jean-Seb just showed you, we can image across a plane in the brain. So now we're looking in the cortex. We found this region that's sensitive using the wide-field microscope, and now we sort of zoom in, right? We try to see what's happening in individual cells. So just like Jean-Seb said, you'll see some cell bodies. It's a little bit more difficult to see, but you'll see sort of these bright spots. The other fluorescence that you see is what we refer to as neuropil, or it's essentially all these dendrites mixed together from all the different cells. It's a more non-specific signal. What we're really interested in are these cell bodies. So again, I'll play you another video. And same thing, the temperature is going to come on. And then you'll see a brief flash here when the cells are activated. Okay? So it's going to start. And right now, you can see some of these cells here in this area getting bright. Maybe. So when you actually run the analysis, what you can do is you can take this imaging plane, and then you can pick out the cells that actually responded to the cold stimulus. And what we can start to do is map which cells are actually encoding thermosensory information. Because ultimately, we believe these cells have to be the foundation of our cold percept. They have to be what's building our representation of the outside world. If we believe the brain is building that construct, these are the cells that's doing it. And so that's what we're particularly interested in, is finding out what information they're actually encoding.
Yeah, but how can we test that, actually, right? So what we can do is we know that these cells will be the cells responding to the temperature, in this case, cold. Now, we want to actually use the mice and ask the mice, I mean, did you feel cold? Or did you feel warm? Right? That's the goal. But of course, we can't speak to the mice. So we have to actually find ways to train the mice so they can feel the temperature and let us know that they felt the temperature, in this case, the cold stimuli. So we've got this brain again. That's the mouse brain. And now we're going to teach the mice to actually detect temperature. And if they do it right, they've got a reward, which is a droplet of water. And they can actually do it. And they're getting really good at it. And they can be really performant. So we measure performance as the mice really report the temperature we stimulated with correctly. Okay? And as the mice doing this, we can deliver the reward. And this is what we call a correct trial. And you see these mice, they actually perform really well. I would like to say like us. They can even detect 0.5 degrees drops, right? All this is done at 32 degrees because our hands actually are not 37, but more like 32 degrees. And now we are dropping the temperature to 10 degrees. So from 32 degrees to 22 degrees. And the mice can feel it really well. But we know that they can also feel a drop of 0.5 degrees, right? Exactly like us. So it's very important to know that this system, the way mice actually report temperature, is probably very similar to ours. Okay, but the basic of our experiment, again, I have to recall you our hypothesis, is that temperature, the representation of temperature of the brain, is in the primary somatosensory cortex. So what do? We take the brain, and we can inject some drugs in this location to block these neurons. And this is reversible, so we can actually probe afterwards and see if the mice recover from it. So we can inject a drug, and we do that here. We inject the drug into the primary somatosensory cortex, and what we found is that the mice can't really perform well. Right? It's below a chance level. Basically, the mice is guessing. Right? The mice can't feel anything. Although I have to say, the periphery, the nerves, they're still intact. So we still will transduce the information to the brain. But that particular region is key to actually represent temperature. Okay. Yes. But now we have the warm stimulus. And then I would like to give it back to you.
Yeah, so everything that we told you so far was about the representation of cold. Something that I didn't make a big point out of, but throughout history has been shown, is that there's more cold spots than warm spots. When you electrically stimulate the skin, you get more cold responses than warm responses. Cold just seems to be more prominent than warm. So once we had some information about cold, we thought, well, let's look at warm. So again, this is the same experiment that I showed you before, the same mouse, the same session. But now, on the paw, instead of providing a cold stimulus, I just provide a warm stimulus. And again, you'll see the video. On the bottom will be the trace of when the stimulus comes on, and you can watch for the response. Okay, and the stimulus comes on right now. Any thoughts? So,
yeah. It's crazy, right? I mean, that's what we thought.
Absolutely. So, across tons of mice, there's no warm response in somatosensory cortex that's reliable. We've searched and searched, let's say.
Yeah, and that was our hypothesis, that the primary somatosensory cortex, which is receiving information from the skin, should be the location of temperature representation in the brain, which is true for cold, but doesn't seem to be true for warm. So, I mean, it's science, right? I mean, often you get it wrong. So what you do, well, of course, you realize you're wrong. That's the first thing. And then you go back to literature, you read. right? You try to see if we found something. And actually in humans, it is known that primary somatosensory cortex, which is the orange ball you see in this MRI there in humans, is important for temperature sensation. This experiment's been done in humans. They've been put into the MRI machine, and they stimulate them with different temperature, right? Warm, cold, and somatosensory cortex, primary somatosensory cortex, is activated. Makes sense. And it's here. You can see that. But what's interesting is that there is a different part in the brain which is activated, which we call the posterior insular cortex. Now, easy to find. It's in between these two lobes. It's quite deep, so you have to really, push the lobes together, and you can see it through. Right? That's the posterior insular cortex, and it's there. Right? It's in between these two in the gyrus there. So then we looked at the literature for the mice, and there is also a posterior somatosensory cortex. We know already that there is a primary somatosensory cortex, and there is a posterior insular cortex in the mice as well. Okay? So from there, we actually did test it, right? It's the same experiment. We've got our mice, and then we stimulate the mice with this time cold temperature and also warm temperature to try to see if this part of the brain is representing temperature as well, which is, was unknown really in mice and how it works at the cellular level. So here we've got the same type of movies that Clarissa showed you before, where you will see this is the population activity of the brain over the posterior insular cortex, and here you've got the timeline, right? So, and the stimulus will happen here. This is the cold stimulus, and you will see the activation. And it's pretty clear, right? Stimulus comes on now, and it's a very, very precise point, right? The posterior insular cortex does represent cold temperature. What about warm temperature, right? So we did the same type of experiment, warm stimulus on the forepaw of the mice. We've got the same setting there. Now the warm stimulus can come in there. Very sharp point, right? And when it's clear, warm temperature are represented into the posterior sensory cortex. So for us, it was mind-blowing because, it wasn't planned. We were thinking primary somatosensory cortex, and we're going to go to the cellular level and understand how the brain and brain cells actually process sensory information, and will give us a clear idea of how the brain represents temperature. But that was actually not the case. Yeah? Okay. Yes. So, of course, this doesn't mean that this area is linked to the perception of the mice. So we have to do the same type of experiment we've done with primary somatosensory cortex, which is inactivating this part of the brain. So we have our mouse brain with the primary somatosensory cortex and with the posterior insular cortex in orange. And what we want to do is this time use light. I told you we can change the genome, we can interact, modify the genome of these mice, and we can express proteins that will silence the activity of the neurons when we shine blue light on them. These tools, they did revolutionize neuroscience, because that's the way you can unbuild the network and see which part of the circuit is actually doing something. So in this case, we silence this posterior insular cortex while we're actually giving a cold stimulus. And what you see there is that the mice has difficulty to actually distinguish between the cold stimulus or no stimulus at all, right? So that's very interesting for us because it means that cold is not only represented in the primary somatosensory cortex, but also in the posterior insular cortex. What about warm? Well, the mice can also detect warm as us, and very, very precisely, right? And in this case, it's also a 10 degrees raise, so it's very easy for the mice to detect, and they can perform really well. But when we shine the light, so when we inactivate the posterior-insular cortex, then they can't actually feel the temperature. They won't get the reward because they can't detect the temperature. Okay. So what we've done today is showing you how the brains represent temperature. And I hope you realize that, it's been quite a journey, and we're not finished, right? What we want to do is go at this cellular level and understand at the cellular level how temperature is represented, right? For now, we've shown you that mice and humans can detect, sense temperature really well. Again, 0.5 degrees drop or increase can be sensed by humans and mice. What we've showed you as well is that while cold can be perceived or represented in the primary somatosensory cortex and in the posterior-insular cortex, warm is only represented or more represented into the posterior-insular cortex, which is, again, a key finding for us. Now we want to use the new techniques we talked to you about, which is two-photon microscopy and all these really powerful tools that will help us understand the relationship between these cells, the way they are interconnected, to get a grasp on the cellular specificity. Because at the end, we think that finding cellular specificity will help us understand pathologies, disease linked to defect in somatosensation, right? This.
Okay, and so the last thing to tell you is that over the last 2,000 years, we've been trying to understand what our skin can sense. And that science, I believe, is a self-correcting course. So while for 1,000 years or so we thought that somatosensation was a single thing, we're back on track now, I think, in believing that it's multiple. And what we present here today is the current state of the knowledge. It's what we believe to be true. And so if you just take one thing with you, I would say maybe the next time you go to grab a beer, hopefully after this with us, that you may touch with your hands, but you feel with your brain. It's your brain that's really creating this percept of the external world. And I think with that, we'll take your questions.
Thank you so much, Clarissa and goodness, I have problems with my tongue today. Jean-Sébastien, for this amazing talk. And again, you can ask questions here in the hall. Please go to the microphone and ask them there. When you talk to the microphone, you have to wear your mask, otherwise you don't. When you go from home, you can ask us questions here. Just open your browser on your mobile device, go to slido.com, SLIDO.com, enter the hashtag of today, BB0522, because we're Berlin Brains, May 22. Type in the questions, I'll see them here, and I will ask the speakers, and they will give you the answer. But beforehand, I have a ton of questions to you guys, so we will have to have a couple of beer afterwards. But first of all, you said it very nicely at the beginning. Neuroscience means studying defects, right? And then interpreting. So we know deaf people, we know blind people, but do we know numb people? Are there people who don't feel, and how do they live?
Yeah, I mean, there is multiple cases, and the one I would like to talk to you about is the phantom limb, for example. People that have lost one limb. So they did lost sensation from that limb, but the brain can still feel what this limb is actually, I mean, the limb would feel if it was still there. And so there is this, this reconstruction from the brain, which is hopelessly using the sensation around us. But when you lose this, right, then it becomes tricky for the brain to really understand what's going on. And it's going to make up some representations that are not actually really representing the surrounding world, but as your experience have built your brain, you will actually try to recover this. The brain will try to recover this.
That is giving me goosebumps. I mean, do you trust your own senses now that you do this work?
I don't think you should trust your own memory. It's all subjective.
Yeah, I mean, I heard this from another talk on jurisdiction, and that the eyewitness testings are always wrong, or people have to ask several people.
Yeah, you
That's
were talking about the blind people. I would like just to jump on that bit, because, I mean, the brain is really plastic. The brain is made to sense our surrounding world. And if you just prive the brain from one sense, these cells that were there for that sense will actually be used for something else, right? And you will get a bigger area for hearing, for example, and that's well known from blind people. They can actually hear much better than other people. And you spoke about loss of sense, but you can actually create sense as well. So you can actually, on your skin, for example, you can build some kind of seeing sensation, and you can train that skin to actually see your surroundings. So if you've got, let's say, a camera which is looking at your surrounding, and the camera will send information to your wristband there, and you've got some kind of tingling when you see lights. Well, now you can close your eyes and then walk around, and then you will see the light, and you can actually go to the light, because you can train your brain.
Yeah, but what's really critical with this too, I think, is for this to work, you need a functioning brain. So there's also the case of cortical blindness. So people who have fully functioning retinas, but they can't see because of a clot or a stroke in the visual region of their brain. So this idea that you don't see with your eyes, you see with your brain, we also see it in clinical case studies, sort of the other side of the coin, I think.
Amazing. Oh goodness, too many questions. First question maybe from the hall, otherwise I have to sort my questions out. Is there a first question here from the hall? Otherwise I will continue with my questions. Either have to speak very loudly and I repeat it, or you have to go to the microphone. I can just go there. Yeah, please. Yes, please. Okay, until he reaches the microphone, let me ask you one more. We talked about sense and temperature, but then you put it so very nicely that sensing warm and cold might be different things. So isn't it one sense to sense temperature, or is sensing warm and sensing cold something different in the end?
I would argue this is one of the biggest debates in the field right now. So there's two competing hypotheses, the labeled line hypothesis and the pattern theory. And so in labeled lines, there's the idea that there is a cold line and there's a warm line. So that essentially these ideas that were developed with back in the 1830s here in Berlin are still true all the way down to a single temperature level. But then there's another theory that says, well, that's not actually true. There's cells that respond to both cold and warm, and they're more of a pattern theory, meaning that the activity or what stimulus identity it was is represented in the activity of the neurons, in their patterns of activity, not just the lines. So I think I would argue that how far to split temperature into separate sensations is an active area of debate.
So but that means that in the end we might have more than the common five senses, right? And temperature is even subdivided in touch, and then proprioception is also something more. Maybe 5 is too low a number.
Depends if how you split it and group it, right? Because vision can also be color vision, right? So you could have 3 different colors, or audition can be multiple frequencies. So it's all how far down you want to go, I suppose.
Please, your question.
Hello. So I actually have a couple of questions, and my first question is actually linked to nerves. So this is a kind of anecdote because it happened back in time, and there was a worker who was working on a building, and he fell, and there's a sharp object that was stuck between his legs, well, his foot, I'm sorry, and then we took him to the hospital and gave him some anesthesis and to soften his pain, but he wasn't actually responding to this anesthesis, and then we actually removed his shoe, and we found that the sharp object was stuck between his toes, which is, and I don't understand why he was crying and yelling of pain. Actually, the thing didn't touch his, make contact with his foot. So how come actually did that happen?
Yeah, I mean, we can trick the brain, for sure.
Yeah.
Yeah. I mean, there's many examples where you can actually trick your brain and stress for things you can't really control, right?
Yeah.
So I would say that's part of it.
And my second question is about the experience with the mouse. So the performance of the mouse dropped from about 100 to 30% something when you injected the drug. But that means that that there is indeed another region where there is sensors other than the primary somatosensory cortex. And does that mean as well that the primary somatosensory cortex cannot work alone because you see 100, so it drops to 30%. Does that mean?
So you mean it doesn't drop to 0?
Yes, exactly.
Yeah, exactly. I mean, I would say nothing in the brain works alone. The brain is highly interconnected. Every region connects to another region, and reciprocity is a fundamental feature of the brain. So the idea that there's two regions sensitive to temperature is not surprising. It's just not something we knew before. No. So for example, in visual areas, I think they've identified something like 17 secondary visual areas. The information really spreads and goes in multiple information streams. So to actually prevent any sort of response, you'd have to, theoretically, I guess, identify every region. But it would be difficult, I think.
Yeah, and then you have to see the experiment itself. So with this task, we asked the mice to feel the temperature, right? So and cold is quite salient. So you really feel it. I mean, we tried on ourselves, and it's you can't really miss it. But sometimes you might. Yeah. And the mice is really a playful animal. So it will try to actually guess. So we can't really talk to the mice, but we can actually pick up the time where the mice start to guess because she didn't feel the sensory stimulus. So 30% is about chance level. So it's like you flip the coin. Not the coin because it's 50%, but it's about half. It's about chance level. So, yeah.
And my before last question is, what happens if the cells of my hand, for example, are damaged? Can I no more feel, or could I actually install something in my hand, like a machine or something, so that could be linked to a nerve or something, so I can feel? Or is that possible?
They want it to be possible. I would say that's the future of sensory prosthetics. Yeah. So as it is right now, like I think you mentioned a little bit about peripheral neuropathy, what happens when you sort of lose these nerves in the skin, and you do lose that sensation. And so right now, I think a lot of the work in these sensory prosthetics is based on people who have lost the sense organ entirely, for example, amputees for skin. But for more common ones, I would say is like a cochlear implant, where essentially that's how it works. So if you're familiar with a cochlear implant, when someone is deaf, you can actually implant stimulating electrodes into the cochlea, which is where pressure waves are transduced into electrical signals. And basically, you have a little microphone, you record what's happening, and then you stimulate the cochlea at different areas based on which frequencies were recorded, and the brain remaps, like what Jean-Seb said. The brain is very plastic. If you give it information, it can learn. And so for skin, I think it's not as developed. A cochlea, I would say, cochlear implants are the most advanced sensory prosthetics right now. But creating sensory prosthetics for the skin is a very active area of research.
One last thing is actually so the weight of the human mind is 1.5 kilograms, and for women is 1.2 kilograms. What is the difference actually? Why is it different? And actually, matter of fact, there's Einstein as well, whose mind is 1.2 kilograms. Wow, how come? What is it? I mean, difference between two genders?
I missed the word. Sorry. Could you repeat the question?
The question was why, on average, male brains are a bit heavier than female brains.
And Einstein, for example, his brain is just 1.2 kilograms.
Yeah, he wasn't
Maybe
really
I can
tall, right?
answer this question because we had it in one of our last shows, and it's simply that on average, female persons are a bit lighter and a bit smaller. And when every muscle is represented in the brain and you're a bit smaller, then your brain is a bit lighter. And when there are more cells to be represented in the brain, then more cells in the brain have to represent it. But that doesn't mean that women are less clever. Definitely
No.
not,
Didn't say
because
that.
that's more the interconnection of the cells, and that's the same in male and female persons.
Yeah, I didn't say that. I mean, matter of fact, Einstein is like, he's a
Yeah.
genius, and his mind is 1.2 kilograms.
If size would matter, then whales would be much cleverer
Yeah.
than
Yeah, I don't know. I don't know about personally about differences in brain weight. I mean, like with Einstein, right? They tried to do a bunch of things in the autopsy to identify what made him a genius,
Exactly,
right?
yeah.
Measure the size of different brain regions, look at the ratio of astrocytes to neurons, and ultimately, I think we don't know. There's also people, I think this is very interesting, doing research on criminals, so serial killers, effectively, and trying to identify what in their brain makes them operate at the fringe of society, let's say.
I see.
Because I think these sorts of things we're really interested in, right? Like, why are some humans geniuses? Why are some psychos? what's sort of linking that together? I mean, theoretically, if you believe what we've told you, everything you experience is represented in your brain, right? So somehow there must be a neural signature of these sorts of behaviors and capabilities, I would think.
Actually, the founders of the institute you're working on, Oscar and Cecilia Vogt, worked on exactly that question 100 years ago. So this is not an easy question.
Yeah, very good question.
Mm-hmm.
Yeah, that's it. Thank you for your time. Thank you.
So I would add something to this, is that, I mean, it's not just the number of cells, right? It's the connections between the cells. It's what we call the connectome. That's what makes us who we are. You know, it's the experience, you know what, I mean, if you take the flies, right? The flies have got a certain number of neurons, and we know exactly what the connection is, and everything is encoded in their genomes, right? For humans, the connections aren't, because there is too many. I mean, you can't just code for all the connections you have. This is only based on your experience, and that's why, you know, babies can't really do anything themselves, because there is no connections yet. And that's the development and their experience that will promote these connections and will influence on, you know, your being at the fringe of the society, maybe. You know, it's what makes you who you are, these connections, and that's quite very, I mean, interesting, I think.
That brings me to another question. You showed this impressive visual thingy with the young lady and the old lady. And I ask myself, when we talk about temperature, are maybe Inuit that live in the cold and Bedouins living in the desert, would they perceive temperature differently?
Yes.
Yeah.
Absolutely. So actually, the proteins that we talked about, the tryptophan and TRPV1, they've actually gotten samples from humans. I think they did Nordic versus, I can't remember, but basically Nordic people had a mutated version of tryptophan. So there's actually differences within our human genomes in the expression of these thermosensitive channels. But I mean, even further than that, I really like to look at like other things. So if you look at other model organisms, we talked about in the mouse, in primary somatosensory cortex, we see no warm. In boas, in snakes, they see no cold. Because snakes are cold-blooded animals, right? And they search for their prey by looking for warm-blooded things. And so they've done tons of recordings, and effectively they're all warm responding. So the needs really seem to match the organism. And so I think I would also expect with humans, there's been things over evolution, but also probably adaptations based on where you live.
You showed this, oh, sorry.
No, I wanted to say that if you're interested in that question and go further, there is actually a specialist there in the crowd, which is Philippe Bocquignac, and would be happy to take your questions there, right? That's actually his topic.
Meet you at the bar.
It's how core temperature affect your temperature sensation, which are two different things, indeed.
And you gave us this wonderful view back into history and talked about Johannes Müller and his law of specific nerve energies. What might he say to somebody who is synesthetic? I mean, in synesthesia, people received, the most famous one was Kandinsky, the artist, and people who are not suffering, they live with synesthesia, means they hear vision, they smell sound. So two senses are always mixed, and Kandinsky was the only one, I think, known where three senses were mixed. So, but this, then what's going on with them?
Must be a mismapping.
Yeah.
I mean, they've done a lot of studies, actually. I think it relates back to this plasticity of the brain type question. You know, they did this one with frogs, where you could take a frog eye and actually physically rotate the eye 180 degrees, okay? So you actually turn it over. And the frog, when it sees a fly, at the beginning, licks down, right? Because its eye is upside down, it thinks this is down. But over time, the brain actually remaps, and the frog can learn to use its rotated eye. So I would guess in something where you view one sense as another, it has to do with some remapping that happened between the sense organ and the region of the brain that's expected to get that type of information.
But we know also that this happens when people do high doses of LSD or psilocybin, then also they sometimes experience synesthesia.
Yeah, might be different reasons. I mean, I would say maybe during development, you might have a defect in regionalizing these sensations, right? This, what we call this receptor field. And then maybe synesthesia is this, right? LSD might be just messing up with your brain, right?
Very well put. Well, what about schizophrenia?
Yeah.
I mean, people suffering from schizophrenia live in a world where they can trust their perception. So is there an overactivity of, what is it?
You know, I'm not sure what it is, but I think it actually relates back to what Jean-Seb said earlier about phantom limb pain, right? Because people with schizophrenia often seem to experience things that aren't happening, right? They have phantom percepts. And similar to people with phantom limb, they no longer have the sensory organ, but they still feel like it's there. They still feel things. And so I don't know what causes it, but I would say I could envision that it's something similar to that you're getting these percepts activated without the external sensory stimulus. So somehow there's a disconnect.
Okay. More questions here. Otherwise, I keep on asking my stupid questions, and you have to hear them because I won't let you out. Yes, please.
Does the hypothalamus play a role in sensing the temperature or something?
For the people at home, he asked if the hypothalamus plays a role in sensing temperature.
Well, not directly, but it's involved indeed, because the hypothalamus is actually sensing the core temperature, which is I mean, clearly the temperature of our body. So if you go outside during the winter, right, it's really cold, your core body temperature will decrease. And then you go back in and you're really cold, and then you wash your hands, and then you've got a sensation of pain, even though the water is not that hot, actually, right? And that's because your core temperature is very low. And that's the hypothalamus's job, is to keep that temperature as the same level, okay? But it doesn't sense, I don't think it does sense the peripheral temperature. I think it's a very different system. Am I correct, I think?
I mean, I think in the thermal pathways, we often think about two pathways, right? One is related to thermoregulation, and one is related to more somatic sensation, like touching things and interacting with them. I think would be fair to say.
Thank you. We have two questions from viewers from home. The first is, why do we have two regions representing cold and only one representing warm?
That's a great question.
We're working on it.
I mean, that's what we're doing.
Another way to say you don't have a clue.
Well, I mean, it's what we're actually working on, really. As of today, it's an enigma for us. I mean, and why do you need a different region when you've got a specialized region sensing sensation from your skin already there? So it's very interesting. It might be due to the valence of the stimulus itself. So what I mean valence is that some stimulus might be nice for you. Some others might be not painful, but not really nice to you, right? And that's the valence. You've got, a positive experience of a stimulus or a negative experience of a stimulus. And it could be that the somatosensory cortex is very important for salient stimulus which are needed to be perceived in order to react quickly. While warm is different, because warm has got this valence of positiveness, right? So maybe somatosensory cortex has nothing to do in this representation, and maybe the posterior insular cortex will actually have a role in this sense. I don't know, it's conjecture.
That makes sense, yeah. More questions from the outside world. Should we assume there are distinct brain patterns for everything that happens? Could different experiences have similar signatures?
Meaning? Can you?
I mean, the latter reminds me of the déjà vu, right?
Yeah.
When
Yeah.
similar things activate some, and I have the impression that I experienced that before. But the first, but I don't know, that's my
I
guessing.
mean, to me, it kind of feels like a question about memory, actually. And the idea that you have, so in the memory field, the idea of how do we form a memory? What is a memory? And one idea is that it is a subpopulation of cells that sort of become activated together. And to say that that population of cells is activated exactly the same way every single time, I think what we know from juries and from our own memories is probably not true. It can be colored by a lot of different things. But for a sensory pathway, we think that there be a more stable representation of a sensory stimulus. Now, whether that maps to a stable percept, I think, is another question. I don't know if that really answers the question.
If it doesn't answer your question, please rephrase your question. Oh, more question, oh goodness. Could each individual have a different brain response depending on development? So how would we ever find one brain pattern for everyone?
I think there isn't. I think that's the beauty of the brain. I think each brain is different. I mean, it's the even how would you say that? Coming from siblings, these that are born together. I forgot the name. The what? Twins. Yes, exact twins. Yeah, sorry. You know, they have the same genes, right?
Yeah.
But they don't have the same connectome. The cells, they didn't really connect the same way, and that's because the experience is different. There is no two individuals that can live exactly the same life, exactly have the same experience. And this experience, these small bits of experience you have, will shape the reconstruction of that image you saw in Hawaii when I thought I was in vacation there, but I wasn't, right? Because you know, we don't see all the same thing.
So what science fiction tells us, that it's able to record your perception and then play it to me as a video at home, and I feel like being Clarissa standing on stage, that's simply bullshit.
For now. I think, you know, this is entirely true, I think, that, you know...
Well, I mean, Elon Musk is building a billion-dollar company on this Neuralink, right? On exactly this idea, basically.
Yes.
Yeah.
Let's see.
Yeah, but I mean, I think what Jean-Sébastien said is exactly true. Every brain is different. But some of the large-scale organization is consistent across people. So the somatosensory cortex is always in the same place. The visual cortex is always in the same place, you know, in the absence of some irregularity. So there is sort of a general structure, but the specificity within that is really unique.
Okay, we have 2 more minutes. Oh yeah, please, say it loud and I will repeat it.
If memory could be triggered by temperature, and if it does, how does it work?
If a memory can be triggered by temperature, how does it work?
Yeah, that's a great question. I mean, that reminds me the Madeleine of Proust, right?
The what?
The Madeleine of Proust. It's a biscuit in France, and in his story, you know, he remembered the smell, and the smell itself reminded him his youth, right? With her mum baking this Madeleine, and I think yes, I think temperature, as we said before, there is this valence, and the memories linked to that softness of warm maybe might bring up some really nice memories where, you know, you were cuddled by your mum, whatever. But I'm sure there is some link, because that's what Clarissa said about memory just before, is that memory is based on the multiple representations, right? Because it's not just touch, it's not just temperature, it's not just a visual stimulus, right? It's all together that forms actually the percept. So yes, you could trigger one memory with one of these single element in that representation, for sure. Yeah.
Cool. Okay, 2 last questions. Oh, okay, please.
I always have been impressed by those magicians walking on burning coal. But I was
Okay, he was asking about the magicians who walk on burning coal or ice cold, and how they trick their bodies to be able to walk on that without burning their feet.
I don't know that trick. So I know many tricks, and I can show you
Complicated
some.
question, simple answer. Yeah.
Yeah, I can show you some tricks afterwards, but that one I don't really know.
Yeah, but I think it's like everything. You can really trick your brain, really. So I think there's a lot of things that have come out lately. Not a lot, but we used to think you couldn't control your heart rate, for example, until somebody did it. They could actually consciously control their heart rate. And so these sorts of things start to make us realize that we actually have more control than we previously believed that we did, for example. So I would guess that the people who are capable of walking across coals either have severe peripheral neuropathy or they've learned to train their brains, right? And say, like, they just create some state where they don't cry, I guess.
With this last wonderful answer, I want to close the Q&A section. We have more questions from home, but guys, when you want to ask more questions, you need to come here to the Urania. And there is the next opportunity for you in just a week. We had to cancel the January event for Berlin Brains due to Corona restrictions, and we will repeat this on the 3rd of June. Come to the Urania and see Susanne Wegmann and Maximilian Frank from the DZNE, the Deutsche Zentrum für Neurodegenerative Erkrankungen, it's always hard to spell, from NeuroCure and the European Einstein Center for Neuroscience. And they will talk about this tau protein that I mentioned earlier, that exists in all of our neurons, and what it does in health, what it does in disease, why aggregate states, something that we don't know from proteins, but it is important, might be important for its function, and why this may lead even to maybe new therapeutic approaches. My tongue is really giving me a hard time today. For diseases like Alzheimer's. So, next week, here in the Urania, or if you're not in Berlin, from outside, urania.de. Thanks for watching, thanks for being here. Stay healthy, stay safe. Looking forward to see you again. Jochen Müller, bye.